So the plasma membrane is a phospholipid bilayer. Y'all are good with that, right? Yeah. Okay. Transport proteins can transport things. That means they also are called transmembrane proteins. To move things across the membrane, they have to be big enough to be on this side of the membrane and on that side of the membrane. Good? And they help things across. That's mostly what this PowerPoint is about. This is how some materials enter or leave the cell, and some of them use ATP. We call that active transport. All right. The plasma membrane is like the bouncer, controls what gets in, what gets out. It is selectively permeable. Please tell me you've heard this. Does this sound familiar? Okay, good. Only some things will get in, but some things will get out. This is a really good example. This organelle, you would agree, is chock full of these dark red, almost black things, and there are very few anywhere else. It's because you're actively pumping. All right, so there are two types of transport, passive and active. Passive, the cell, I should add to this and say no energy needed by the cell. Okay? If the cell is not spending any of its own energy, it's passive. If the cell has to spend energy, it's active transport. You're putting molecules in a concentrated area. Did we do the Japanese subway thing already?
Yes.
Okay. Then you remember the example. I don't need to do it again. You understand that if you have to move things from an area of low concentration, like the subway platform, to an area of high concentration, like the inside of the subway, it might take some energy. It might take 5 people with like gloves amount of energy, right? I feel sorry for that kid. All right, so there's the example. Passive transport is when things are moving in the direction they want to go. We've got a lot of glucose out here and not so many on the inside, so things want to go this way. That would be passive. We have lots of these little red doohickeys here and only 2 there, so passive. Moving from where it's high to where it's low. That is like opening the door on the subway. You don't have to tell people to get off of that Japanese subway. They want off. Active transport is you do spend energy. You move things from where you don't have very many to where you have lots of them. And the same thing in the other direction. You have lots of the green ones here and not as many green ones here. So in there. And so what do? You pump things that way. Takes energy. So diffusion is passive transport. Diffusion is the movement of things from a high concentration to a low. They want to spread out. They want to move apart. And it'll stop if you get everything the same on each side of the membrane, inside and out. Once you have things equally spread out, there's no reason to move because life's not any better on the other side of the membrane, right? All the molecules want space, all the ions want space, and there's no real benefit if it's the same on the other side. Now, there are things that cause diffusion to happen faster. We did this in lab last week. Heat is motion. Heat is energy, right? So say heat is energy that causes motion, and because of that, if you heat something up, you'll cause it to diffuse faster. Also, bigger differences. How many people, I saw a few people had their diffusion labs out. How many people had more change in the bag of pure syrup than they did in the bag of water?
Yeah.
Yeah. You know why? Water, you have water inside, you have water outside, there's not going to be that much motion, right? But if you have pure syrup on the inside, you're going to have motion into the syrup trying to dilute it because water wants to dilute things, right? Now the next question, how many had more movement into the pure syrup bag than they did into the half syrup bag, the 50%? Tell me why. It's because a big difference makes things move quickly. The water wants to move fast. For the movement of the water, we say osmosis. For the movement of everything else, we say diffusion. The goal is always the same, though, to get to equilibrium, to get everything as diluted as possible. Okay, so small things diffuse faster than big ones. Bigger differences cause bigger diffusion. Heat changes causes an increase in the rate of diffusion. We're good with all this. You've seen this, right? All right. Little things pass through phospholipid bilayers easier than big things. and especially non-charged, non-polar ones. Carbon dioxide goes through really, really easy. Oxygen goes through really easy. You take in oxygen, you use it, you give off carbon dioxide. They just pass right through the membrane. Jeremy, what's the problem with this?
Polarized.
Exactly, it's polar. This is hydrophobic. It hates polar things, especially hydrophobic. It hates water molecules. So how does water get across that? We're saying this moves through simple diffusion, or in this case it would be simple osmosis. How can that get across?
The membrane's always moving.
Yes! Okay, these heads can move around a little bit, right? So most of the time, you're not going to be able to get things across. But every now and then you get a water molecule that's moving fast enough, they'll just hit the membrane and bump those phospholipids apart a little bit and zip right through. Now you're not going to be able to bring 20 water molecules across because that's a big charged group and that doesn't work. But one or two can hit hard enough to slide it apart. Hydrophilic things go through a little, but hydrophobic things pass through really well because that feels like home, right? That's easy. So we talked about osmosis, moving the water. Water wants to dilute stuff. Like I said, osmosis and diffusion, we're basically talking about the same type of thing happening. We give a special name to osmosis. Osmosis is the movement of water. The solute is not moving. Diffusion is the movement of the solute, but the water is not moving. In both cases, we're trying to make things as diluted as possible. Good? So in lab, we talked about hypo and hyper and isotonic solutions. Tonic means concentration. Iso means same. Good? Hyper means above. Hypo means below. So if we have an isotonic solution, it is the same concentration as the inside of the cell. If we have a hypertonic solution, it is more concentrated at the inside of the cell. If we have a hypotonic solution, it is less concentrated than the inside of the cell. Does that make sense? What direction does water move? Well, you know, water wants to dilute stuff. Water is going to flow towards the concentrated. So in isotonic, you do the same amount of water in and out. In hypertonic, you're going to have the water move out of the cell into the more concentrated solution, right? In hypotonic, you're going to have water move into the cell towards the more concentrated interior, because water always tries to dilute stuff. Now, this is the book drawing. I think you saw this already, right? Just remember, water wants to follow salt. I'll tell you right now, there is a very specific, very tricky question that you'll have to read carefully, very carefully, on the next test about hypo, hyper, iso. Read it carefully. Now, this is the drawing I was trying to make in lab, because you know I'm a big fan of the smiley face. The smiley face works on my periodic table, so it works here too. All right, when we have, and basically this is good because it shows the difference between animal and plant cells. All right, animal cells really love iso. When we have an isotonic solution, we got the same amount of water going in and out. That's true for animals, true for plant. And animal cells love it. Plant cells, they can tolerate it, but it's not as good. They're not as happy that way. Now, in hypertonic solution, the cells are losing water. Water is flowing from the inside out to the more concentrated because water wants to dilute things. That's going to kill the animal cell. That's going to kill the plant cell. They both think it's a bad thing. The reason why is a little bit different. The animal cell here, you see the shrinking, and now you see these things sticking out, poking. How many of you have ever been to the mall? And you go through the mall around Valentine's Day, and you see these things where they put a rose inside a balloon, right? What would happen if you let out all the helium, let out all the air? And it starts to shrink down, and the rubber starts to get stretched over the thorns of the rose. Would you agree you could pop the balloon, right? Punch holes in it? That is the same thing that's happening here. These are the intermediate filaments sticking out. They're part of the cytoskeleton. And as the membrane is shrinking, it is going to get holes punched in it by the things inside the cell. Good? Make sense? Here, we have the same thing happening where the water's flowing out, the cell is shrinking, cell is shrinking. But what you're seeing is the cell's attached in a few places. In reality, plant cells are a little different than most animal cells. You got a plant cell here, a plant cell here, a plant cell here, and they actually are attached not just to the cell wall, to the other plant cell. There are these little things called plasmodesmata, like little tunnels between cells, so they can transmit things from this cell to that cell. It can't shrink away. It's stuck there. So what happens is, as the vacuole loses all of its water, this membrane tries to come in here, but it's attached there, and you get a tug of war that will stretch the membrane until it tears. So here it tears because it's getting a hole poked in from something inside. Here it tears because this can't shrink and the rest of it can. So slightly different reasons, but in both cases, if you throw it in a hypertonic solution, you are going to punch holes in the membrane. There's a fancy P word for that. We call it plasmolysis.
Oh, I see.
Plasmolysis, it lyses the plasma membrane. If you put it in a hypotonic solution, the water rushes in. Do we agree? The animal cell will inflate, 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 inflate. So do we agree? Hypotonic solution, the water flows into the cell.
Yes.
Yes. Okay, and the cell tries to expand.
Yes.
When an animal cell starts to overexpand, it's not happy. If you expand it too much, it will pop like a balloon, spill its guts, and die. Good?
You're gonna have that time.
Yeah, a real bad time. Now, a plant cell is a little different. A plant cell has a cell wall made out of cellulose. Okay, so when we expand, the plant goes from here. This is the plant in an isotonic solution. And then you water it with a hypotonic solution, and it perks right up, right? All the leaves stem. It's happy. Now, some cell walls are really thick and really tough, like the ones in wood. You know, wood, strong. Some cell walls on other plants are not quite as thick. And I mean, you know, there's some plants that are pretty flexible, and it's better to think of their cell wall as being like Lycra bike shorts. You do realize that they provide support, that you can fill them up real big. You do realize you can also overfill them until they fail, right? You can do the same thing to cell walls in some types of plants. Are you with me? All right, moving on. So, this is turgor pressure. We talked about turgor pressure before. If we have so much water flow out that we tear holes in the plasma membrane, we plasmolyze it, this cell is going to die. Do we agree? Plants. If it's flaccid, that we have enough water flow out that the plants live. If we have enough water flow into the vacuole, fill it up, it perks up, we say it's turgid. Yes, the same words, turgid and flaccid, you can laugh and giggle now, same words as a penis. It means the same thing, okay? Flaccid, it's not full of fluid. In case of penis, blood. In case of the plant, it's water. Okay, turgid, it's full of fluid. So we're going to talk about transport. This is just like one of those. Here's a bulleted list we're about to go over. We're about to go over all of these. Sounds like we've done most of them. All right, passive transport. You agree no ATP is being used, right? Okay. So, little things, we already said this, little things, uncharged things, and occasionally water can cross with no problem at all, right? But if you're big, if you have a charge, you need help to get across. You need a transport protein, one that expands the membrane, and there are two types that we're going to mention. We're going to talk about channel proteins, they're like a tube. We're going to talk about carrier proteins. All right, facilitated diffusion. Facilitating means helping. We're helping the molecule across. But it is still passive because it is still diffusion. You're still going from a high concentration to a low. Here we've got very concentrated, trust me, I know it's a snippet of a bigger picture. We've got lots of these, whatever the triangle depictions are. We don't have as many out here. So, it's diffusion if you go from high concentration to low. but they can't cross the membrane easily. So what do? We put a protein here that's a channel that they can pass through. Are you with me? Okay. Now, the cell is not spending any energy to move these because they naturally want to spread out. They naturally want to go from high to low. That's why it's diffusion. These channels are limited by the size and the charge. Would you agree that if I tried to take a molecule this big and pass it through that channel, it doesn't work? Right? Well, anybody in here know why I don't want to eat pufferfish?
Poisonous.
Yeah! You know anything about the poison?
It's like is it a neurotoxin?
Yes, it is. Very good. Yeah, it's a nerve toxin called tetrodotoxin. All right, so there is a sodium channel. Why is there a sodium channel with sodium small? Because sodium brings water with it. Enough water, 2 molecules, that it has trouble making across the membrane. That makes sense, right? There's a channel. Good?
Yes.
All right. There is tetrodotoxin that tries to pass through the channel. You are not surprised that it will not fit. No one's surprised by this, right? So why is that a problem? That alone would not be a problem. It would try to pass and it would float away. The problem is that, and I don't remember which one's positive and which one's negative, but the mouth of the channel has a certain charge and there's the opposite charge on the tetrodotoxin. So when it bumps, it sticks and it plugs the channel. It's like you corked a bottle and nothing gets in and nothing gets out through that channel. And since nerve conduction depends on sodium going into the nerve, if you cut that off, you cut off nerve stimulation. You cut off the ability of the nerve to send a signal. You no longer get a signal from the brain to the diaphragm saying, breathe now, dummy. And so you don't, and then you die. That's okay. I've been bitten. Ow, he has teeth. Ow, ow, ow.